Hello, Biochemistry 1 students. Welcome to our enzyme webinar on alpha galactosidase. My name is McKenzie Cheynet.
And my name is Jessie Bronski.
All right, let's get into it.
So first, we're just going to give you guys a brief overview of what we're going to discuss today. And the first thing we're going to discuss is the background of our enzyme, just to give you guys some basic info before we get started on anything else. We're then going to look at the sequence, the structure. We're going to do a little bit of bioinformatics application. And then we're going to look at a specific disease that's caused by a mutation in our enzyme, and that's Fabry disease. And then we're going to look at our primary literature review, and we're going to discuss that.
So first and foremost, just a little bit of a background on alpha galactosidase. The one we will be primarily looking at today is found in Homo sapiens or humans, obviously. The UniProt ID is found below if you want to know exactly the protein we're looking at, but its main function is to catalyze the hydrolysis of glycosphingolipids and participate in their degradation. Now, it makes sense that it's part of the hydrolase class of enzymes since it's found in the lysosome. So hydrolase means that it's breaking things down. Lysosome, that's where things are degraded. We have the common names listed, and if you see GLA in any of the upcoming slides, that's the alpha galactosidase gene we are referring to. And we have the enzyme number below as well, 3.2.1.22.
So this is just a overview of our sequence. It's 429 amino acids long, but we're going to look at the structure in its folding. So it has a mass of 48,767 Daltons. It belongs to the glycosyl hydrolase 27 family, and that family is known for catalyzing the hydrolysis of glycosidic linkages that result in hemiacetals or hemiketals, which we know as reducing sugars. It's also part of the signal domain, and it is known for being present at the N-terminus of proteins, and that can either be in the membranes or they can be secreted, and they're very important in signal recognition. And both of the subunits, as we can see, there's two main subunits, and they are identical.
Next, we're going to get into the bioinformatics application side of things, and we'll be using UniProt to do so.
So the first thing we did with UniProt is we created possible primers for our enzyme. As we can see here, UniProt gave us an amino acid sequence, and from there we went to the European Nucleotide Archive and we found the nucleotide sequence for our specific protein. It gave us an mRNA sequence, and we converted that to DNA. And for the forward primer, we copied the first 20 base pairs of that nucleotide sequence. And for the reverse primer, we got the reverse complementary of the last 20 base pairs.
Next, we're going to get into the binary interactions of alpha galactosidase A. Binary interactions are essentially direct physical interactions between proteins via van der Waals forces or hydrophobic interactions, and they help to form a signal transduction pathway. So the first protein we have listed here, GDP-MAN, obviously mannose. I'm not going to attempt to say the rest of the protein's name. And we have its gene in the parentheses here. It's involved in the assembly of dialcohol-linked oligosaccharides. These are important component of N-linked glycosylation, and N-linked glycosylation is an important part of our protein's function. Next, we have sortilin. It's important because it transports proteins from the Golgi apparatus to the lysosome, and since our protein is found primarily in the lysosome, obviously it's going to be an important interaction.
So we ran an alignment and we found similar proteins. The first three are the Bolivian squirrel monkey, the orangutan, and the night monkey. And those three share about 90% of their identity with the alpha galactosidase in humans. This is just a different version of alpha galactosidase in these species. And then the last three rats, mouse, and zebrafish only share about 50% of their identity with the alpha galactosidase that we find in humans.
So next we use Clustal to run a multiple sequence alignment. And essentially how we did that was we used the align function in UniProt. We got the sequence of alpha galactosidase in Homo sapiens and the sequence of alpha galactosidase in the Bolivian squirrel monkey. And we align those two against each other in Clustal to get this multiple sequence alignment. Now, we love it because the different colors show some of the differences in the amino acids, and we have boxed the different amino acids or the differences in the amino acid sequences. Hopefully, we found all of them. If we didn't, we're sorry. But for example, at amino acid 115, there's an arginine instead of a histidine. And you can go through some of the differences if you would like. Next, we're going to go into Fabry disease. Now, this is the most prominent disease that you're going to hear when you hear alpha galactosidase. It's a rare X-linked sphingolipidosis disease leading to the accumulation of glycolipids in tissues. It happens when glycolipids are not being hydrolyzed or degraded, so obviously they're going to accumulate. It leads to chronic pain, vascular degeneration, cardiac impairment, and more. And since it's X-linked or alpha galactosidase is X-linked, it affects primarily males, and it's going to affect 1 in 40,000 males.
So the primary literature that we chose is called The Catalytic Mechanism of Human Alpha Galactosidase. It's by Goose and Clark and many others. And if you want to find out who those others are, they're going to be down in the references. So in the introduction, they describe what is known about alpha galactosidase already. And we know that the job of alpha galactosidase is to remove the terminal alpha galactose residue off of polysaccharides, glycolipids, and glycopeptides. But it is also an alpha retaining enzyme. And what that means is that both the substrate and the product are going to have anomeric carbons with alpha configurations, which means the mechanism has to take into effect that it's going to be anomeric at the beginning and it's going to be anomeric at the end, and alpha for both. So the other main thing that was known is the extinction coefficient, which is 18.1.
Awesome. Next, we're going to go into the previously proposed mechanism before Goose and Clark's research. It's a double displacement reaction mechanism, which basically means there's two consecutive nucleophilic attacks on the anomeric carbon, which leads to the retention of the anomeric configuration. It requires two carboxylates, one that attacks as a nucleophile and one that attacks as an acid or a base. And looks like here is our acid or base. And the electronegative fluorine atoms come in at the 2 and 5 position, allowing the reaction to slow, which allows for the trapping of a covalent intermediate. And this is that electronegative fluorine, if you can see my mouse right there. And this previously proposed mechanism has two cooperative active sites, and cooperativity, is what Goose and Clark are primarily going to be studying.
So to get these results, they conducted many steps to get a variety of results. The first step was they looked at the molecular biology. So they found that the human alpha galactosidase could be expressed in insect cells. From there, they took the GLA gene, as we described above, and replicated it via PCR. The final product of this step provided an alpha galactose sequence with C-terminal histidine tags to be able to identify them in future steps. And they also created mutants that were named D170A, and these are mutants of alpha galactosidase. That brought them to step 2, where they infected insect cells. They transfected them with insect medium, which allowed for higher transfection, plasmid DNA, and Helimax, which creates liposomes to introduce the desired gene, the alpha galactosidase, into the cells. They added blastocidin 3 days later, which is an antibiotic that inhibits protein synthesis. So they stopped protein synthesis from there. And that brought them to step 3, where they wanted to express and purify the protein. And they did this by looking for cells, the insect cells that express the mutant D170A alpha galactose protein, and they grew them for 3 days. They substituted the binding agent for a nickel binding buffer, which purified, so it killed anything but the enzymes tagged with His. So the His tagged recombinant proteins. From there, they ran an SDS page analysis to find the pure alpha galactosidase. This brought them to step 4, where they wanted to look into the kinetics of the enzyme. And they did this by monitoring the alpha galactosidase hydrolysis of a synthetic substrate called PNP-A-gal. And they did this at an absorbance of 400 nanometers. They started with 1 microgram of the enzyme, and they put it in a citrate phosphate buffer and added this compound to 8 different substrate concentrations of the PNP-A-gal. From there, they recorded the absorbance every 10 minutes to tell the difference between the solutions. And then that brought them to step 6. In step 6, they crystallized and ran X-ray data collection to be able to get structures of the macromolecules. And they did this by obtaining crystals of aspartic 170 alpha gal mutant. And they hang dropped them via vapor diffusion, which is a method for crystallizing macromolecules. And from these crystals, they made atomic models of the different steps of the mechanism.
Awesome. Next, in the first part of the results, they formed four crystal structures that represent the 4 stages of the catalytic mechanism. The 4 structures are seen here. We have empty with no substrate, substrate attached, enzyme substrate intermediate, and lastly, product formed.
So from there, they looked at the covalent, they looked at the steps, the mechanism, and they found that the first step was fast. And they found that because the leaving group was a really good leaving group, so the first step was fast. The second step, however, as we mentioned above, the covalent intermediate has to be trapped in order for this mechanism to work. And to do this, the second step has to be slow so that the intermediate can be trapped. They also found, if we look at the bottom, that there are 3 ligand conformations. So the ligand underwent a conformational change from C1 to S3 back to C1. And one of the last conclusions they made is that they confirmed that it was a homodimer, which means it has 2 different active sites. However, they found that the active sites were not cooperative. They found that each monomer has 2 domains, an N-terminal beta-alpha 8 barrel, which contains an active site, and a C-terminal anti-parallel B domain. So they both had active sites. They determined that the first active site, however, only selected alpha anomers, and they determined this by putting a mixture of 70 to 30 beta to alpha anomers of galactose and found that only alpha were bound. And they also found that the second active site only binds beta anomers. So in this image here, they constructed a diagram of the second ligand binding site, which only would select B alpha anomers, and it has trypsin as its primary molecule, amino acid.
Lastly, in their discussion, they found a few rules regarding the structure of alpha galactosidase. So the first rule being that the enzyme must align the syn lone pair of the nucleophile so that it can attack the anomeric carbon opposite of the leaving group. And this has to happen to achieve the inline attack of the nucleophile. Their second rule was that there has to be very little distortion in the substrate. And the third being that the substrate conformations can be used very differently. There are many conformations that the substrate can take on depending on the type of sugar, such as S3, C1, S2, S5, and many more.
And then this is our references slide. So as I mentioned above, if you want those other names on the primary research paper, those are going to be found on this slide and any other resources we use. Thank you guys so much for listening. I know that was long, but.
Awesome. Thank you guys so much.